tell you you're live now.
Excellent. So last week we talked a bit about how we talked about an overview of synaptic plasticity, learning and memory in the brain. So one what I wanted to do last week, but we sort of didn't have the I didn't have the time to, or got carried away with, was how does it this actually fit with continuous lifelong learning, uh, aka how does the brain avoid catastrophic forgetting? So this meeting, I wanted to recast the stuff we discussed about in this context, and I wanted to get your guys' input for ideas. A lot of this is still either vague or in the hypothesis stage for in the literature, and then some things are well known, but a lot of it isn't. So a sort of overview of what I'll be talking about uh, is, for example, how do different plasticity mechanisms? Can you guys see my mouse?
Yeah.
Okay, good. 'Cause I don't need to use a weird pointer thing. What do the different developmental and plasticity mechanisms contribute to avoiding catastrophic forgetting? So, developmental patterning, for example, changes. It adds a certain specificity to what neuronal populations can see and stuff. So there's a sort of segregation, and also during patterning, you have synaptic competition.
Aries, I don't
And
know what this, I'm not sure what this term means. Are you going to go into it? I don't know what developmental pattern
oh,
is.
sorry, yeah, right. So this is just generally what happens during development when the brain
In
area.
utero. In utero.
Yeah.
So how does that relate to catastrophic forgetting?
Well, generally the idea that the fact that you have this separation of input and output populations, such that, for example, if you look at a fully connected neural network, it could be, you might be exacerbating the fact that it's fully connected. You might be exacerbating catastrophic forgetting by the fact that all inputs and outputs have like all neurons get input from like everything else, for example, and you have a certain specification that could allow you to overcome that partially. Then you have this sense of competition in sensitive periods. Apparently, they don't call them critical periods anymore. By which you could sort of adjust. You can adjust what inputs you're sensitive to, and sort of do more of this separation, for example. And again, the idea is pattern separation and completion are mechanisms that are well known to exist in the brain, and how do these contribute precisely to avoiding catastrophic forgetting. And then in the more functional side, I wanted us to discuss how internal models of the world might help doing this, in the sense that, do we learn in terms of existing models? Our memories are memories just new things? Do we have to like write new synapses and be like, okay, this is memory A, this is memory B? Or is everything learned in relation to everything else, and how might that help with continuous learning? And by the way, I suspect it's the second option. So I just wanted to, I'm not going to go over the details of this. I hope you guys remember some of this from last week. But the idea is that there are these gradients during development that guide axons, and there are these pruning processes that happen, such that neurons, that axons find specific targets, and that their connections are meaningful. So here, for example, we have axons from the retina extending into the superior colliculus, and there's what we call topographic branching, which occurs due to these chemical gradients. And then you have arborization and competition locally, such that only interesting inputs, only interesting axonal projections are kept in a certain population. And of course, this happens in the cortex too. These gradients lead to interesting gene expression patterns, which segregate areas functionally. So basically, the theme here is, maybe it's a bit naive, but the theme is here, you have a selectivity in what any given area of neurons processes, basically. And this gives you both the segregation, an anatomical segregation in this case, of how things are processed, and also it also gives you new modules in which you say like, oh, maybe, you know, how learning occurs is that you have the activation and the involvement of different types of modules, which gives you a sort of, in a combinatorial sense, allows you to create richer and more stable to perturbation memories.
But I mean, this is an argument for segregation of function, but when I think about catastrophic forgetting, I guess maybe I'm always thinking in terms of the brain and not in terms of neural networks. But you still have the issue. This alone doesn't explain how you avoid catastrophic forgetting, because you can now experience catastrophic forgetting in the visual system, or the auditory system, or the somatosensory system.
Of course, yeah.
It's just you're just segregating the problem, but you haven't addressed the problem of catastrophic forgetting. That's how, unless you were saying something else.
Oh, okay. Sorry. So I started this by assuming everyone knew what catastrophic forgetting was. And yeah, in this case, you can have, like Jeff said, within like a certain population of neurons, you can have catastrophic forgetting because you know, new learned things can overwrite old ones. Basically, that's what catastrophic forgetting is, and then you can't retain old information. I wanted to take like a global approach and start like, okay, you know, the fact that you don't have, there isn't, you know, the cortex of the brain isn't one homogeneous layer that just receives input from everywhere, and then you, which would more easily lead to this kind of
Yeah, but I think from a neuroscientist point of view, they would never think of it like a neural network. They would say, okay, within any subpopulation of the brain, we have this issue. But just because you're segregating into different functions doesn't really address the catastrophic forgetting issue. You still say, oh, I could have catastrophic forgetting in this area, this area, this area. So
absolutely,
yeah, it just doesn't really address that issue. It maybe most neural network researchers wouldn't think of it this way, perhaps, but.
Right, yeah, and the idea of this is to sort of see like, how does the brain do this differently than neural networks? And of course, you know, if you have many different modules that are separated across the entire cortex or the brain that deal with any one given thing that is learned, then the fact that these areas are anatomically separated and will give you like more capacity, for example, in a naive way. So this is just there in case, and as a review, if anyone forgot this, you know, you have both synaptic potentiation and depression, and potentiation occurs when, you know, there's a certain, in the Hebbian sense, there's a coactivation of pre and postsynaptic partners, and you also get depression based on, you know, the timing, the frequency of activation, and these all lead to to gene expression changes that cause permanent structural change to the cell. Right. So I wanted to talk a little bit about competition and critical periods, because in this theme, so you guys can tell if this is like redundant and we're not addressing the point as much, you guys can let me know. But basically, you know, this is the work that gave Hubel and Wiesel the Nobel Prize. Was that here what you're seeing are ocular dominance columns. So this bright patch here is something, this is in visual cortex, and here they injected, in one eye, they injected a tracer basically, which gives this white looking, this bright signal. And you can see that there's a patterning that says that every, you know, every local area has a preferred eye that it gets input from. So, you know, within an area, as you said, Jeff, you have this sort of segregation of inputs. And the only way that this would work is if you have a certain competition between different inputs and input statistics. And here on the lower panel, you see this, what happens if they remove one eye or suture it, is that the other eye ends up dominating the input space here. So you have this this plasticity, and there's different plasticity mechanisms that you need to explain that. So not just basic Hebbian correlative plasticity. This is one example. It's called the BCM rule, the Bienenstock-Cooper-Monroe rule, and basically this has a dynamic threshold where basically Y is the output of the neuron. And there's a the threshold for plasticity is this here, which is also updated with the synaptic weights. And this is the synaptic weight, such that imagine this curve sort of moving right and left, so that when you have competition, the threshold you need to reach to overcome your the neuron you're competing with for input is much higher, for example. So this is a way in which you need to supplement Hebbian learning, which Hebbian learning would by itself lead to catastrophic forgetting and other problems. It would lead to rampant activation, for example, and catastrophic forgetting in the sense that like you would overwrite correlated synapses with new correlated synapses. And you need, if you have these thresholding mechanisms in this competition, you can avoid that to some extent.
Hey, Aris, is this something that the K-winner rule also, K-winner take all, also kind of implements, that the more activity there is, the higher your threshold has to be in order to win?
Yeah, I wasn't gonna talk about our stuff or stuff in neural networks, but yeah, it does do something similar to this. One thing that this doesn't have, of course, for computational, for the ease of computation, is that the competition is not specific. And here you also want to have a specific competition between correlated inputs, so that one of them or very few of them win.
Okay, so you compete specifically only with inputs that are correlated with you.
Yeah. So if you think that Hebbian learning works based on correlation, in a naive sense, and yes. So what you'd also have is something like, you'd have some K winners because you have inhibition and stuff in the network. And then you'd also have competition between those K winners so that you further sparsify your activity. And this is similar. I'm not going to go into this. Sorry. So another thing we talked about on Wednesday was homeostatic and three-factor plasticity. So you can easily imagine how homeostasis can help you with catastrophic forgetting, in the sense that you don't want, you want to you want your activity to stay within a certain range, and your plasticity to stay within a certain range, and as such, you don't want to create too many new excuse me. You don't want to create too many new synaptic associations at any given time. So the brain probably initially did this as a sort of energy conserving mechanism, but it helps in catastrophic forgetting in the sense that not only do you sparsify associations, but also you sort of avoid overwriting of old ones, since you diversify what inputs, you diversify your weight updates. Does this make sense?
Yeah,
And
I'm still maybe, we're thinking about the term catastrophic forgetting differently. I mean, this one definition is like, oh, the whole system fails because you've forgotten everything. That would be truly catastrophic forgetting. That's what maybe neural networks would do. But here again, we're just by distributing and sparsifying and so on, you still have a fundamental problem of how neural networks learn without forgetting old stuff. And you can break it up into pieces, or you could say, I'm not going to learn something now. But that doesn't, to my mind, that doesn't really get at the core issue. The core issue is, how does a neuron learn something new without forgetting something it's learned before? Of course, we have a particular proposal on this. So to me, these are all mechanisms which sort of dance around the outside of the problem. It's not, you're not really getting at the fundamental issue that when you form, if you have a neuron and you modify like a classic point neuron, like you might have a neural network, and you modify its synapses, you're going to be forgetting other things. And that's the fundamental problem. So to me, these are all this is more of a discussion about distribution and sparsification, but I don't know, am I getting this wrong? I don't want to get you off track here, but it seems like none of these things are really addressing the fundamental issue of how does a neuron learn something new without forgetting something else. We're just saying we're distributing around in different ways and have different mechanisms for preventing neurons from learning continuously. but that's not quite the same problem in my mind.
So you could take this down. Homeostasis occurs at the level of a single neuron too.
Yeah, but if the homeostasis says, "Oh, I'm not going to learn all the time because I'm trying to maintain something," well, then you're avoiding catastrophic forgetting by not learning something. The trick is how do
you're allocating different resources to learning than you.
Yeah, but once you can allocate them differently, but you still have the fundamental issue underlying it, which is neurons have to be able to learn something new without forgetting something they've already learned. And you can say, "Oh, I'm going to allocate someplace else," but then that other place has to have that same problem.
Well, then, effectively, if you saturate, if a neuron only had like 2 synapses, that would be a huge problem, right? You could one association on synapse A and one on synapse B. Neurons have many thousands of synapses, right? And if I do think that this is the fact that you have synapse, you can retain synapse A, and you can maintain synapse A and learn new things on other synapses. I think within the same synapse, if you override it, if you learn a new association, that synapse will be probably
overridden. Obviously, we have a bias about this, because our paper, why do neurons have 1,000 synapses, really was addressing, in some sense, this very issue, and it required 2 things. It required compartmentalizing the learning on the dendritic branch of a neuron. That was the key element, so that you could learn on one branch of a neuron and not affect the learning on another branch or another segment. And to be that's like fundamental. And if you didn't do that, then you're not going to achieve continuous learning, or you're not going to achieve forgetting. And the other one was the way neurons get reused in different sparse distributed representations. So the same neuron can be used in many different representations because you're looking at a collection of them, a unique set at any point in time. So a neuron can respond in many different contexts to different things, but in combination with other neurons, it's always unique. So it was those 2 elements combined that allowed the system to basically incrementally learn new things without forgetting anything else. And we demonstrated that many times with the sequence memory. You could learn 1,000 sequences, and then you could learn 1,000 more, and the original 1,000 weren't forgotten at all. So those are the elements. And I guess I'm sensitive to this because it felt like, for many, many years, this is one of the issues I grappled with, which is like, how do neurons learn, learn continuously and not forget things? And that was the only answer that we came up with. Everything else, these other issues that you're talking about here, seem to be sort of secondary. They're like, you can segregate and sparsify and do all kinds of stuff, but until a neuron can learn a new pattern and not affect an existing pattern it learned, which required segmented learning in the neural in the dendrites, you just couldn't achieve this. That's the way I view this.
I run the risk of changing the topic too much, but just briefly, we're talking about a specific kind of continual learning, or a specific one of the problems of continual learning is that or one way to frame it is that we want to be able to learn new associations, form new synapses without disrupting old information, old knowledge. That is one way to approach all of this that covers some problems. But I think another part that also comes up in HTM context, like old school HTM, is as your basis changes. If you think of neural networks as learning a basis, encoding the sensory input in a basis, you want the basis to keep improving without forgetting old things. The equivalent in HTM, old school HTM, would be like, as our spatial pooler learns, how does the temporal memory stay functional? Like, how does it keep working as the spatial poolers change? And I would say that, like, in my mental model of deep learning, that question I just asked with spatial pooler temporal memory changing bases is almost the harder problem. How do you, because as you're training the neural network, you want it to keep just getting a better and better basis that allows better and better classification in general. And we're mostly focused on the first part of, like, not disrupting existing synapses. And I feel like we're leaving
it's not clear, but
which is fine. But
it's not clear that brains are really good at the second one. This is what, you can't teach an old dog new tricks thing, right? We form this basis set, the spatial pooler output early in life. And the way our temporal memory works is if you were to redo that, you are going to forget those things. You're going to forget your sequences or your additional learning. And so, and I think there's evidence that humans aren't good at that. That we really aren't able to do that very well, and this is why young people come along and they understand new things better than old people, because people all the learning is based on this previous. So I would argue that the plasticity of the spatial pooler is much lower than the plasticity of the other half of the temporal memory. So I'm challenging whether brains can really do that.
It's a good point. I would say that I'll end it with this, or at least I'll try to. I would say that brains have some mechanisms for addressing that, like maybe replay is helping that. Maybe there's some cognitive things, maybe dreaming things cause you to re-invoke old representajust
learn them again, right? It's sort of like saying you just have to relearn these tasks in some sense. If you're as your body grows throughout the first 20 years of your life, in some sense, the basis function of your somatosensory representation is changing. And I can't cite any papers on this, but my belief is, and I think there's evidence for this, is that the things you learn, how you learn to do things when you're 5 years old, you have to relearn them all the time. Like you're not keeping that memory. You just have to practice again and again and again until the basis stops changing, because you just can't transfer that knowledge. That's the impression I have.
But just a concern here. So you're making good cases here. My concern is if we're gonna try to make deep networks better at continual learning, I worry that the bigger problem, the harder problem, the one that it would be more impactful to solve is the second one with the changing basis, and the disruptions to existing connections might be the smaller problem.
Maybe I don't know if there's a solution to that. I mean, maybe I'm going to get out of my area of expertise here, but imagine if I take a system that's designed to recognize images, okay, and the analogous would be okay, I've trained it on natural scenes, and now I'm going to train it on unnatural scenes. Well, your performance, whatever that is, your performance may not be as good as if you trained it initially on natural scenes. But it's not clear that you'll be able to. How am I going to say this? I mean, you may have to live with the original basis, and you have to figure out the best job you can do with that. It may not be possible to take a vision system that was trained on one entire type of problem and then have it continuously learn into a new one without creating problems. I'm not sure that's possible. It might be, but I would argue that it's not possible from a biology point of view, and that you're going to have to learn to live with it. So I think it'd be great if we can do it, but let's not set ourselves unrealistic goals here. Clearly, if I had a set of images and we classified 1,000 images, 1,000 types of categories, and then I came along later and I said, okay, I'm going to take that same network and now learn a new 1,000 categories. If there's a lot of similarity in the basis between those two, then I should be able to do that. If there's not a lot of similarity between them, then I don't expect it to do that. Just maybe leave it like that.
If I may add something, so this is what I had the Held and Hein paper for, the Kim Carousel thing. So I would argue that along Jeff's argument, the fact that the real organisms, the real brains, they don't just look at one thing naturally, right? We create a basis set based on our interaction with the world, and the world has certain statistics and a certain behavior to it. And then when we learn new things, we're basically recomposing, we're synthesizing our existing basis to create, let's say, in a feed-forward net to create a new higher level representation, for example. So our basis probably, this is something that these sensitive or critical periods are probably very related to. It's to learning sort of the base, let's say, statistics of your environment. So in this experiment here, and there's been a lot of experiments like this, by Heldenheim is called the kitten carousel. It's very famous in psychology class.
By the way, just so you know, we've talked about this experiment a bit. So I don't know if everyone knows it, but it's a lot of us. It's not like we're unfamiliar with it. So you can summarize what your point is about it.
Right. Yeah. And the argument here is that these kittens, they were raised, they were raised in the dark room, and then the first thing they ever saw was this environment with only vertical stripes. So there's one other famous experiment, which is like if kittens only see vertical stripes for a certain part, they cannot see horizontal stripes. They just run into things, or stripes of other orientations. But this, because these kittens are young, they can learn. If you put them in a natural environment after this, they will quickly recover. It takes them, I think, 8 hours or 24 hours to recover. And in this experiment, basically, there's one kitten who's walking around the carousel, drove optic flow, right? So when the kitten moved, the stripes would move with it. And it learned this association, the sensory motor association. Whereas this kitten was just in a little box and was passively viewing the stripes go by, dependent on the other kitten's movement. So these kittens over here, these would like have severe sensory motor deficits for 8 hours because they could not learn they didn't learn that their movement is coupled to their environment, for example. So you could see these as types of bases, right? Like these basic statistics, like when I moved, the environment was with me. When I perform an action, my paw or whatever was going to be at this part of the world, it's going to move things. And this works as well. And this also extends to the types of Gabor filters and other associations you might learn early on. So I think these bases are largely invariant in some sense. So we could argue about how they form, how much of it is plastic and how much of it is just the statistics of input and the fact that you have inhibition, etc. But generally, I would argue that the one of the ways that brains do this is that they synthesize new input. And in a neural network, if you train it on, let's say, the GSC data set, it's only seeing these statistics. And then if you want to learn like the MNIST or fashion MNIST or something, you have to rewrite or add new bases. So one thing you would do, which is very hard to do, arguably, is to have it behave more like a robot and move around and learn bases in an unsupervised manner. And then my intuition is it would be better at learning to classify new things it hasn't seen before by synthesizing these bases it's learned. And yes, Jeff, you are right. This isn't so much about catastrophic forgetting per se, but about like continuous learning. So I'm not addressing the specific catastrophic forgetting aspect as specific.
Okay, I just was reacting because that's what you said that.
Yeah, you're absolutely
today's topic was. So.
Yeah, you're right. Yeah, what I'm trying to get is lifelong learning, and catastrophic forgetting is obviously a part of this. So, right, yeah, here we just have neuromodulated Hebbian plasticity, which means you can add a gate to a correlation whether or not you want to learn it, and you can imagine this furthers your capacity, et cetera, et cetera. So I'm going to go into internal models now. This is going to be very short, but I want to ask if anyone has any like suggestions or questions or opinions about just basic plasticity mechanisms before that.
I have one. Several people have mentioned when you're talking about long-term changes to the synapses, and they keep referring back to genetic expression. Genetic expression would affect the entire soma. So I'm trying to figure out, you're trying to lock in a particular activity at a particular synapse on a particular branch. How can you relate that back to genetic expression?
Right. So you mean in the developmental phase or in the genetic expression during gene expression during plasticity?
Any of which.
Any of which. So during plasticity, this has been a very big problem, the sort of, let's say like a synaptic credit assigning problem. And there's been several ideas of it. I think there's been some insight, but I'm not very sure what it is. I haven't read those papers thoroughly, but the idea is that, let's say, imagine this neuron, you have multiple other synapses on it, right? So you learn an association between the pre and postsynaptic activities, such that you strengthen the synapse, right? So there's short-term potentiation, which means that like locally, the synapse will traffic more excitatory, more glutamate receptors. I mean, the postsynapse will traffic more glutamate receptors, and generally this synapse will become more responsive. But this gene expression, which is a long process, will drive the synthesis of new proteins. But these are, as you said, these are driven, these are trafficked around the entire cell, right? They're not only driven to the specific synapse. Is that your question? Like, how does this?
Yes.
Yeah. So they're driven to all kinds of other synapses too. So the idea has been for a while, and I don't know what new evidence has occurred in the past 5 years or so, that, well, imagine that this is, whatever this is the protein that's being trafficked to a synapse, and this could be any synapse. That if these synapses are to be potentiated, that there's some molecular factor here that is changed or released locally that affects the configuration of this quote unquote plasticity protein that's been distributed everywhere. And thus only the ones that would, only synapses that should potentiate will lead to the structural change, because these proteins will be affected or drive transcription locally, translation locally at the synapses that need to be potentiated.
I'm sorry, when you say translation, what are you referring to in that case?
Sorry, so transcription is the chain, reading DNA and creating RNA. Transcription is basically activating gene expression. Translation is when you create proteins, mRNA and proteins. So translation, so if you have local translation, you can have like an mRNA or something, and you can locally create proteins because you have like a Golgi apparatus everywhere in the cell. You can have a Golgi apparatus here, and you can create these proteins locally. So
I was under the impression that was the main mechanism people proposed, that the protein synthesis was local.
Localized protein synthesis.
Yeah. So yeah. Is that still? That was my impression. I think that you're saying the same thing, right?
I think so. Yeah. That's that's been the sort of most attractive.
So the cell basically says, "Hey, I need to grow something here," and it starts generating more of these messenger RNAs. And then when the messenger RNAs reach the correct spot in the cell, there's other factors there that say, "Yeah, convert this into proteins here," and we'll build something here. But it's based on some other molecular marker that was left around from the synaptic activity.
Exactly. Okay, but you have gene expression. That's either, in my mind, is either saying this thing is always cranking out these RNAs, so they're hanging around waiting for an opportunity to create a protein in response to some stimulus, or it's not. It
might be both. It might be both, Kevin. But it definitely, there's a lot of moment-to-moment gene expression, like which genes are active in a cell is constantly changing throughout the life of the cell based on situations. So it's not like it's set up once. The genes are being turned on and turned off by various factors, and it produces more of this protein or less of that protein. This is a continuously variable process.
So let me just try to tie it back. If we're saying that, I'll pick a number. Let's say we have 1,000 synapses. If some percentage of them are sending out some kind of characteristic signal such that the nucleus says, okay, now I need to express more of something to kind of snap the connections over here or something like that. So it sets up a global mechanism that is activated because for some reason the cell determines, yeah, I've got to lock in something. Okay, I can kind of understand that. But has that been characterized? The other thing is to know there are some organelles that move around, and I've seen pictures where mitochondria travel down the dendrites, sometimes due to pathological conditions, but they're capable of moving there. So I'm just wondering if there's anything mediating this thing rather than
there might be. This is a really complicated field, and I think we have not paid attention to it because these mechanisms aren't really that important for the functional aspect of this. But I believe there's a lot of literature on this. And if you want to, you can dig into it. But, from our point of view, that is a functional point of view, what's important is that you can have this localized structural plasticity, and it can be based on various things, but the actual molecular underlying point, how it actually occurs, which molecules and which, RNA, and it's interesting, but it doesn't really, it's not that, as far as I can tell, it's not that important to how the whole system works. So, but there is a lot of literature on this. So if you wanted to, you could dig it up. But I've never felt personally that it's been that useful. It's more useful to know that there's a structural plasticity, and it can be very long term. And it's not just like increasing the concentration of some molecule someplace. You really do grow and form new things and structurally build things.
Okay, so when we refer to genetic expression, that's basically, we're saying, synonymous for some sort of long-term process.
Yeah, I think that's when Ari started up by saying, were you talking about during development or during the life of the, and we're talking now about during the life of the organism, that these gene expressions are happening.
Oh, okay. So okay, on a long time scale, I could see, different gene, okay. So
yeah,
right.
Things are being turned on and off dynamically all the time.
Yeah, I wasn't sure what the time scales were that it was being referred to here, because it seemed like
talking. I think this is even minutes and hours.
This is hours. Yeah.
Yeah.
Many hours, like a day.
Okay. Well, all right. I'll go and pursue it on my own then.
It's interesting if you want to be a molecular biologist, but go for it.
I think it can be interesting for our stuff, in the sense that new ideas come up in this field frequently, and there's sometimes some things that you wouldn't have thought of that change the underlying properties of learning. That you can only get an insight to this if you investigate it from this molecular angle. And because it's harder to do experiments that manipulate synapses in a very specific way. But yeah, again, the current knowledge, as far as I know of it, is not, it's sort of a layer of abstraction too low for.
Yeah, and there are, and by the way, I do believe there are channeling mechanisms, and I don't recall what they are. I think I've read this, that there are mechanisms which channel and direct certain things from the soma to certain parts of the dendritic tree and not the other parts. So there's all kinds of screwy stuff going on inside of a cell, and it's really, really complicated. But in the end, you just have a synapse that either it becomes more permanent or structurally more stronger, or you extend the length of the dendrite or things like that.
There's one aspect of these sort of molecular things that could be important for continuous learning, and I'm not sure if you were going to go into it, Aris, or not, but the idea, in our models, we have this notion of permanence, where, some synapses are just going to be really, really hard to unlearn because they're important for some reason. And, one of the ways that could form is through some, infusion of some neuromodulatory effect, like you hear, something is really painful, or, you burn your hand on a stove or whatever, and you just sort of imprint those memories in such a way that it's just extremely hard to unlearn them. And I don't know if you were going to cover that or not, but that certainly affects, some of these issues of continuous learning and preventing forgetting.
Yeah, the thing that was striking me was when he showed that neuromodulated example where you had a third input to change the characteristics of it. I guess my interest in it was basically saying that if there's these mechanisms around, is there some emergent behavior that not from an organism point of view, but from a mechanism point of view, that is yet another contributing factor to learning permanence and whatnot, if the mechanisms themselves have an emergent property that allows you to have yet another modulatory contribution, maybe at a larger or smaller scale. But that's kind of where I'm going from. It's not so much I want to dive down to and look at the molecules, but I'd like to look at the structure to see if it suggests something emergent coming out of it.
So I just to remind some people who haven't been around for a long time, this idea of permanence, I stole from a paper, and I don't remember what paper it was. But the author was looking at the effectiveness of synapses, and synapses themselves, the amount of neurotransmitter they release at any point in time is rather stochastic. They're not very reliable. The number of those vesicles you can see here in that picture in the upper half of all the synapses on the left here, those vesicles, they don't always work and they're not always around. And so the author was observing there's this huge variation in the amount of synapse, neurotransmitter that's released on any particular synapse. And therefore, the idea that you have some sort of weight to the synapse is kind of a fabrication. They were saying, big synapses can release a little bit of neurotransmitter, and small synapses can release a lot under different conditions. But what the author then argued, they said, what it looks more to me like that the important part of what a synapse is not how much neurotransmitter it releases, but its permanence. And that maybe we should start thinking about the permanence of a synapse, like how long-lived is the synapse, is the important part of learning. And whatever this paper was, and I don't remember who wrote it or whatever, but it really struck me as a very important observation. And that's where the term came from, because that author had used that term. And so I just giving you this sort of context behind this, that sort of flipped my thinking completely about how to think about synapses from like, this sort of scalar or floating point weight thing, which we see in neural networks today, which we're trying to get rid of. But that is a total fabrication, and that doesn't look like at all in brains. Brains are really synapses. The reliability of them is very, very poor, but the permanence is what matters.
So the reliability is a complicated topic, because if you in steady state conditions, the neurotransmitter release will be variable, but it will be enough. There will be let's say a binary operation by which it will be able to like trigger enough activity in the postsynapse, for example. By what I mean with steady state is that when a neuron is active over a period of time, its vesicle pool with the neurotransmitters gets depleted.
Yeah, I understand that. But this author was making even an additional argument. It was saying even if you take that into account, that yes, there's a momentary depletion. So whether you're having a train of action potentials very quickly changes it, whether they're spaced out. The author was going beyond that, saying even beyond that, there's a large stochasticity to the thing. And I'm like, I can't make the argument because I don't remember the paper, but I think it was a good point. It's not to argue that there's no variability in the strength of a synapse, but the author was making this point that even if you take into all these accounts, it seems to be pretty unreliable relative to what most people are thinking in terms of modeling.
Right. Yeah, so that's true. And I think mechanisms like pattern completion or redundancy in synapses might have a role in this, but I'm not an expert in this.
Anyway, it got me to believe, all of a sudden I flipped it around saying any individual synapses cannot be counted on at any point in time to do anything reliable. Therefore, you should always have a population. You always have to be doing a population encoding. And then, of course, later we learned about NMDA spikes and how they work, and it made it all made sense at that point in time. So now that any individual synapses can be unreliable. If you have 20 of them in a row, then they're on average, it's going to be quite reliable, the pattern recognizer.
Yeah, and just because with Subutai asking about permanences and stuff, yeah. So in our models we have this this variable, permanence, where you can have like it be 0 and there's a threshold, right? So this sort of exists in real synapses, in the sense that weak spines, weak presynaptic, postsynaptic elements have a bias towards like being more easily primed to grow, for example. So there's a scaling factor. And once, I don't know exactly about thresholds, but when they grow, they're more reliable in triggering.
That came, by the way, that idea came from Dmitry Tchaikovsky, I can never pronounce his name right, who sort of invented the idea of this potential synapse. And he argued that an axon and a dendrite that are near each other, that don't have any connection but are close, are a potential synapse. And he made the point that when you have the right sort of even just sort of Hebbian type of alignment between these two, they will start to grow a synapse, even though there isn't a synapse. So Hebbian synaptic plasticity doesn't rely on the fact there's a synapse. It just relies on the fact there's an axon and dendrite next to each other, close enough, and through a mediation and perhaps a glial cell, that just you have a pre and post synaptic activity, it'll start to grow, and and so you have some period of which the synapse is growing, and you have to keep reinforcing it before it has any actual effect. So that's where this idea of a threshold came along. It was his invention, that you have some amount of growth that occurs before the synapse actually does anything. Which I think was a brilliant idea. It was just like, oh, that's brilliant, and so that's where that idea came from. So there's a lot of bias. There's a lot of evidence for that idea.
Oh, so there is. Okay, I was going to ask that. Yeah,
it's been shown like when the synapse first forms, obviously there is no synapse. So why does it form? It's not just random. It's literally a synaptic. I mean, there is a Hebbian type of plasticity that occurs between an axon and a dendrite that has no synapse. And it starts to grow a synapse. These little microphilia, they're called, I think. They're really like string-like, growth of a synapse. And it keeps growing. If you keep reinforcing it, it grows and grows and grows until it actually forms a real synapse, at which point it starts releasing transmitter. So that's where we went from this the synapse model, which you see in neural networks today, which is like a continual variable, to one where it's like, no, it's a binary effect, but it's a continual growth or permanence factor. So we have a scalar permanence, but a binary, which I don't, real synapses aren't binary, but they're close to it. That's a good way of modeling stochastic synapses. You can just call it binary. And so, don't assign any, but it's that population growth. Anyway, that's where the whole neuron model came from. There's a lot of evidence for this, and these other people's ideas.
Jeff, did you say that might be mediated by a glial cell?
Yeah, there's a lot of evidence that says even an axon and a dendrite that are too far apart to form a synapse. They're just, synapse, a spine could be a few microns tall, and you can have an axon and a dendrite. They're further apart than that, much further apart than that. There's like, and yet through Hebbian learning, there could be a glial cell in between, and the glial cell acts like a matchmaker. And the glial cell says, hey, you know what? You two ought to get together. And I don't know the mechanisms. I think they're known, but I don't know them. The glial cell can basically encourage the axon and dendrite to move towards each other so that they're now close enough to form a synapse. So the idea that this potential synapse pool is much larger than if you only count places where the axon and dendrite are really close enough. It broadens it out to a much larger area where the axons and dendrites are not close enough, but they can get there if the glial cell says, you guys want to get together.
Well, that's satisfying.
It is. It's sort of elegant. It really opened up the whole concept of between learning as occurring just in some weight change in a synapse to learning forming new synapses of this huge potential pool. And that is such a more powerful way of learning than changing weights. It's like you have a huge number of neurons, and you can go from 0 to 1 versus a limited set synapses versus a limited set of synapses. You can incrementally change its weight, which is a much weaker form of learning.
Jeff, if you, I'm Googling this, but I can't find related enough things. If you have any of these papers at the top of your head, could you email?
I don't have them at the top of the head.
I can send you some, Aris.
Awesome, thank you.
Thank you. Yeah. You'll quickly learn as you have learned. I cannot remember papers at all. So I'm serious. I can't. So
I think the Shklovsky paper is a good one to start with.
Oh yeah, well, that's the basic.
Yeah, it's 2004, so there might have been a lot more since then. But even there, they say, rewiring can occur up to several millimeters distance, which is a huge distance if you think about very drastic situations.
Is it that much? Oh my God!
In case of they're talking about in case of lesions or really dramatic change, it can go. Several millimeters.
That's right. You're right.
Would you CC me as well, Subhatak, please?
Yeah. It's all cited in our neuron paper. I'll send it out to our distribution list.
It was interesting. I talked to Dimitri a while ago, and he's now working at some research institute. It's like a financial company or something like that. Anyway, I think he left somehow he left his research, what he was doing before, and he had not continued to pursue the potential synapse idea. I think he was disappointed in how few people picked up on it. So I don't think he's been doing any more research in that area.
Okay, I found the Chlavsky and Svoboda paper. I'll do some more reading on this. Thanks. Right. So, do we have any more questions or observations on plasticity? Okay. So one thing I wanted to get into is internal models, and I really like this idea that you learn things. And as we talked about the basis functions we learn things in relation to things we already know. So this is one of my favorite visual illusions. I don't know if you guys can see it through, but as you move your eyes around, you could you go blind to some of these circles that are there at the vertices. And this effect, this could be a retinal effect, but you also get this effect when you're far away enough that your fovea encompasses a large part of the image. So my suspicion is that there's a pattern completion at play here, where your brain sort of fills out the rest. Is like, oh, this is like this very strong pattern in the image. I'm just going to fill the rest of the visual field with this pattern because I don't really care. So there's this visual illusions are one of the major contributing factors to the idea that we have internal models of the environment. So the question is, how do internal models manifest? How could they manifest? Because we don't really know much about this at this stage. Sorry.
Sorry, I'm going crazy over here. How many black dots I should be seeing in this picture?
How
many black dots are there actually?
So there's
47.
there's one here, one like every two.
I think there's
It's a
12.
4x3
Yes,
grid.
12.
There's
Yeah,
12
there's
of them.
12. Yeah.
Oh, that's
There's
a really
12.
good
There are only-
visual illusion.
Yeah, but you
Okay,
can only
thanks.
see a few of them at any given time.
Okay, thanks.
So
It's hurting Michelangelo's eyes as well as mine.
sorry.
It's hurting Michelangelo's vision as well as mine.
Okay, so I'll just change.
I think
the slide.
there's a very easy explanation for that.
What is your explanation?
Because obviously wherever you fixate, you see them, right? And where you're not fixating, your spatial resolution is very low. And so to distinguish the little circle from the lines requires a very fine spatial resolution, and it's just not available to the brain at that point in time. So it's dominated. I mean, you could call it filling in. I don't think it's the right way to word it. It's just dominated. The peripheral vision is dominated by these big structures, the big lines, and what it requires to see the individual dots is a fairly high resolution spatial part. It's just not available. It's just not coming in from the retina at that point.
Well, you could try this by moving really, really close to the image.
Yeah.
And sort of increasing your.
But the high resolution spatial part of your retina is extremely small.
Right, but it doesn't die off immediately, right? It's not a delta function. It does get worse the further out you get. But near your fovea, there is some spatial resolution, which, you could see, if you're fixating on, let's say, the top right, you can see, if you're close enough, you can see like the nodes, the vertices, nearby, like outside that.
You can see the one or two or something like that.
And if you
mean the circles or the other things?
The other things where
I
things at the
them.
resolution of the circle.
I don't think you really see them. It's again, that might be like the model argument that you're having. You're basically assuming it's the same everywhere.
Well, if you move far back and you can fit more of this image in your fovea, you move far back enough where like most of the image is in your fovea. And yeah, you balance that with the fact that you could still, discern the individual circles where you're fixating.
But then when you move far back, then you also lose it. If you ever come to my house, I'll show you a piece of art my wife made using film canisters, and it's fascinating. It has the same effect. It basically, there's an image there, and you cannot see the image because there's these little circles that are dominating the spatial structure. But if you blur it, you see the image. It just pops out immediately. It's a dramatic effect. It's almost like
that's awesome.
this. And I'll have to show it to you. But anyway, I'm going to stick with my belief about it. I think this particular illusion, if you want to call it that, has to do with spatial resolution of the retina. I don't think.
I'll argue with
could
your
be.
debate. I'll argue with that. That we can see color on our periphery, but you could make the same case based on what you're saying that we shouldn't see color on our periphery because we only have cones in the fovea.
No, I'm not saying you don't see anything in your periphery. I'm just saying to pick out that little circle requires high resolution, spatial resolution in your peripheral.
But to pick out color in your periphery.
But if I had, let me finish. If I didn't have this background grid, I think obviously all those little circles would be obvious because it's the only input. And you would see them all. You'd see all 12. If I didn't have this background grid, you'd see all 12 all the time.
Well, I could never see all 12 all the time.
No, if you didn't have the background grid.
Oh.
If it was just 12 dots on the screen, you'd see them all. It's the fact that you have competing inputs. You have these very large spatial structures that are competing with a very small spatial structure. And I don't think it's worth arguing over it. I think it's interesting optical illusion, but I don't think I'm just arguing. I don't think you should read too much into it.
Right, but this competition. Sorry, this competition is like a form of the pattern completion. One could say. But anyway, yeah, there are other illusions that can demonstrate different levels of this. So the question is, how do internal models manifest, and how do the predictions they lead to manifest? How are internal models updated, and how can they be used, or are they used to synthesize new information and learn concepts in relation to existing concepts? So one of the earliest that I saw evidence of internal models was in a review paper where somebody was arguing for the extreme feed-forward grandmother cell idea of visual processing. And this is something that happens in a fusiform face area, which is an area in let's say your visual system that is very selective to faces. And this is this was an fMRI study, and they showed different faces. And you might have heard there's like this concept of a mean face and like an average face. And we tend to be attracted to people who have like the closest to the average face. This is just an interesting factoid. But you could create an axis here of distinctiveness and identity. Identity as you roll, and distinctiveness as you go away from the center here of the mean face. And neuronal activity seems to be very well explained by this. As you shift around the circle of distinctiveness and identity over here, you get your neuronal response sort of segregates along the roll, and it becomes stronger along the radius. So basically, you recognize faces as a deviation, as a specifically, like coordinated deviation from the mean face, for example. And then some really hacky study showed that there's correlates of predictions in the visual system and the prediction errors. This was a joke. This was my paper. And one thing that I thought was really cool was I only sort of realized this when I read this paper, was that you synthesize knowledge. So you could see things like a segue, and you could imagine a lot of things that are sort of similar to a segue, and you could take the fact that there's wheels on a segue and something that looks like a handlebar and stuff, and you can recognize it being like, oh yeah, this has the components of things I already know, right? And then you could do the other thing is that you could combine this with like other things in this category of wheeled things that move around and create an entirely fictional object. And you can imagine many different ones of these, right? So there's imagination has a role, of course, and there's a very interesting thing to be said about synthesizing. You call them templates or whatever you want, priors. So the idea is, how does this work? Well, what happens in the brain that allows this to happen, and how does this facilitate continual, lifelong learning? Do we have like a semi-static pool of, bases that we learn during, development when we're young, and then we can sort of put them together and form arbitrary things? And how does learning if you were placed on a totally alien environment as an adult? Let's say you go to a parallel universe where the laws of physics are different. How much would you actually see, right? Like how would you make sense of anything? And this is one of my favorite ever studies. And this is basically biological motion in triangles, for example. And not only can you recognize biological motion, you can attribute attentions and even theory of mind to them. You could be like, oh, this this is like a mother triangle or something. This is like a child triangle, and this one doesn't want. I think this experiment was, this little triangle doesn't want to leave the room because it's scared or something, and then the mother triangle is encouraging it to leave. It's bananas that we can attribute such high level intentions and even emotions to these things, because we could very easily see biological motion. We're sort of wired for it, but the visual statistics of this resemble nothing from what we've experienced ever in our lives, right? I mean, other than cartoons, for example. So you could show this to somebody who's been raised in the forest, has never seen civilization. You could show them this and they would recognize this. I'm like 98% sure.
But the temporal dynamics are certainly recognizable, right? That's the point of it.
Exactly, right?
Yeah, it's like a very simple analogy to this is you learn a melody in one key, and now you play it and hear it in a different key. Well, the actual patterns coming in your ear are completely different. There's not a single tone that's the same, and yet you recognize it's the same melody. So it's the temporal dynamics that matter in many situations, not the actual spatial pattern.
Right, but we know the spatial, yeah, exactly. The temporal patterns and the motion and all that are.
So if you accept that, if you accept that notion, then it's not so remarkable. It's remarkable. It tells us that. It tells us that the temporal dynamics are what's important in this particular case, not the specific imagery at that point in time.
Jeff, if what you're saying, then, if those triangles were replaced with two dots, you'd still get the same emotional impact?
If you could, well, there's two things here. There's a large one and a small one, and there's a direction to them. So, like, obviously in the triangle, the big triangle pushes with its narrow end. So if I just said two dots, I lose some of that temporal dynamics. I can't specify that. But yeah, I think you could. If I had a big circle and a little circle, different colors, I think you'd still see it.
So the spatial relationships do have an impact then?
It obviously has an impact, but the reason you can recognize this without it is that the temporal dynamics. I think if you had a big circle and a little circle, you would see the same thing.
Yeah, well,
but they're intrinsically spatial temporal dynamics, because if you had the same temporal dynamics in things that didn't make sense to you, like there's an enclosure, if you had like random shapes moving around that had the same behavior, it's not clear that you would identify this. There's cues that prime you towards biological motion.
That's why I gave the example of the melody. The melody has no clues like that.
Yeah.
And yet you don't even notice a different melody. You don't even notice it's in a different key. You can't even tell unless you have perfect pitch. So that's one of the reasons we studied sequence memory early on, because it was forcing us to think about the temporal dynamics as a key, because almost nobody thinks about that in vision, right? None of the neural networks, or hardly any of them, now there are some, but in the beginning, none of the neural networks dealt with it at all. And so it's a key part of touch, it's a key part of vision, it's obviously a key part of audition, and yet historically it's been ignored. Less so now, but in the past, absolutely so.
Right. I mean, it's also been very hard for a long time to investigate it. But yeah, sure, I agree with it.
Yeah, I mean,
I would
that's why we studied it.
But I would not consider temporal dynamics to be something that occurs independently of spatial dynamics.
No, of course not. But the point is, clearly they're both important in almost everyday life. But there are some times where spatial dynamics are the only thing that matters, like static image, and other times where the temporal dynamics are the only thing that matters, like melody. But most of the time, it's a combination of both.
Sure. So, right. So with this, I wanted to bring us to a discussion, if we have the time. If people want to throw ideas around, how does this stuff work? How do predictions and prediction errors drive learning? How do you create learning and segregation? Let's say you have a prediction error, I want to predict this thing. How does that create a new knowledge? How are internal models represented, in the brain? Are there different components? Super naive old school idea would be there's there's spatial resolution components in different parts of the visual system or whatever, temporal, and then you bind them together somewhere, and there's all these different modules that represent different aspects of something that's happening out in the world. How does the brain actually do this, and how do you synthesize knowledge? How do you easily recognize things you've never seen before, and how do you imagine things that never exist? And I do think, and is this relevant? How relevant is this for lifelong learning? So yeah, with that, I'd like to leave the discussion to you guys.
Can I start by asking a very naive question? Isn't this what the thousand brains theory is about? I mean, how internal models are represented.
That's right. It's not a naive question. It's it's exactly right. Yeah, I think it's you know, the concept that we've come up with of a cortical column being a complete sensory motor system is really the most concrete form that I'm aware of how neural models are created in the brain. And of course, it was borrowed from the hippocampal complex, but that's right. That is what the thousand brains theory is about. Right,
of course. But, you know, casting this into, of course, the thousand brains theory addresses this as well, and with its HTM forefathers. But, you know, how does lifelong learning fit into this, right? And again, you know, we don't need to restrict this to the thousand brains theory and HTM, but like other evidence we have as well and other potential models like predictive coding or what have you. You know, how would you, like, based on what we know, how would you synthesize, other than the thousand brains theory, or we could elaborate on that, how would you put things together such that you learn continuously based on your internal model of the world?
Well, it's an interesting question to put it that way, Aris. So, I mean, in some sense, this is how the whole theory came about. We're trying to understand these questions, and there weren't sufficient theories out there. Mm-hmm. As far as I'm concerned, predictive coding is not a sufficient theory at all. And you look into it and you see all its deficiencies. And so in some sense, you know, at least in my research and what I want to do and what we have been doing is trying to understand how this occurs in a cortical column, in that sensory motor model that occurs in the cortical column. So it's a legitimate question to ask: how does continuous learning and lifelong learning occur in that model? And I'm not saying we shouldn't look elsewhere, but I don't know of anything elsewhere that's even come close to it. That would be, you know, I'm open to it, but I haven't found it in all these years.
Well, yeah, I mean, predictive coding is an incomplete theory, right? It doesn't have it doesn't have all these things implicit in it. It's sort of it's probably true to an extent, but it's probably within a larger framework. Yeah. Something possibly like the thousand brains theory.
I mean, and even the thousand brains theory, it's driven by the learning is driven by prediction errors. It's not top-down prediction errors. It's moment-to-moment prediction errors. The internal model itself makes predictions, and it doesn't come from someplace else. And so, instant by instant, it's predicting every second. It's predicting what its input will be, and when it has an error, and the temporal memory paper goes through the basic mechanisms for how this would work, how it is that those errors, the prediction errors, what the model does to try to, you know, to compensate, how it learns when it has an error like that. It says, I predicted X and I got Y. What do I do about it? And so we've spent a lot of time studying that. But it's not the same as what most people call prediction, you know, predictive error models.
There is evidence. I mean, it's mostly from Gary Keller's lab, but there's a lot of this in fMRI too, where, you know, you see that there is good evidence that, you know, predictions can be long range, for example, not occur within one column.
Yeah, no, totally. And so, you know, you could think of our voting mechanism as a type of prediction as well. It says, you know, some parts of the brain says we're seeing a dog, and it's telling everyone else you should be seeing a dog, and that is like a prediction to those columns. So it doesn't say that thousand brain theory doesn't say that the columns are independent and that they don't communicate. It just says that you can understand all the processes involved within a column. So prediction doesn't occur, you know, someplace that's always long term or long distance. It's local primarily, and it can be communicated distance wise if you need to. Well, it does almost all the time, but it's not relying on those long range connections. You know, this idea that there's prediction errors are propagated down the hierarchy. That may be true, but it's also true to say that even if I don't have a hierarchy, I can make predictions and learn from them. So it's basically sort of trying to I mean, if either you believe in the common cortical algorithm, if you don't. If you believe in the common cortical algorithm, then all these things must happen in the cortical column. They can't rely on something happening elsewhere that then this one column uses it. Then there'd be a differentiation going on.
So one sort of semi-argument would be, is there a necessary level of redundancy here? Like, would it be more computationally efficient if you could, if let's say, long-range projections, if you have long-range projections at the receiving end of the feedback projections, sort of calculate statistics or represent things in a statistical way, like conditioned on what the different feedback inputs are, for example. And they don't necessarily need to be models of entire objects, but sort of, oh, this is like this sort of thing that happens that co-occurs with this this long, when this long-range projection is active.
I didn't follow that. Maybe.
Let's say you don't necessarily need to learn entire models within a cortical column in V1, for example. Let's say like the ACC or the retrosplenial cortex or V2 even sends a long-range projection to a cortical column in V1 and says, I have this sort of higher, more abstract, let's put this in a hierarchical way. I have this more abstract, fuzzy prediction of something I'm going to see. And you don't need to know about objects specifically. You need to know about the local statistics of part of an object.
Yeah, okay. So that's basically rejecting the concept of the common cortical algorithm, and which is where 99% of neuroscience is today. I remember talking to Bartlett Mell about learning in V1, and he studied that, and he said to me, "Oh, we don't believe V1 learns anything. It's just a filter. It doesn't learn anything. It's just this filter that picks out features, and then it sends it on to someone else to figure out what it is." And I found this astounding because V1 is one of the largest regions in the neocortex of almost any mammal, and to say it's not learning anything was just amazing. But that's where that idea progresses to. It's like, oh yeah, V1 doesn't need to know anything. It can't. It just seems it's a dumb filter, and someone else is doing all the heavy work. Well, that could have been a good design for a brain. It could have been.
Absolutely, and I'm not sorry. Maybe it wasn't clear. I'm not trying to argue anywhere near in favor of that. I do think that V1 can learn models of objects or even more complicated things like associations. But the fact that if you want to like split your computational work such that you don't have like an unnecessary degree of energy expenditure, so that columns and parts of the brain.
The question is, if every cortical column is doing at some level of description the same thing. I mean, obviously they're not doing the same thing. The face region is doing something different than an auditory region, right? They're not doing that the same. But there's some functional areas, some functional description of what a column does that's the same or very, very similar everywhere. It's not going to be exactly identical everywhere, but it's going to be close. And so if you accept that idea, then you're forced to think about this problem to say, well, okay, fine. So somebody else is thinking back and sending something back to V1, but V1 has to be doing something similar too. What is it that it's doing that's the same thing as that other region? And how are they're differentiated by some other mechanism? So to say that, okay, somebody else is doing the heavy work and V1 is doing, some light work, that sort of goes against that principle. And this is why, you might argue, okay, well, that, Mountcastle was wrong. Perhaps you think that's right. He was wrong. These columns are doing something different. But there's overwhelming evidence that they are doing the same thing. So, this anatomical and other arguments we go into. It's a reasonable starting point, and that's where Hubel and Wiesel started from, and we've been living with that model for 60 years. But that doesn't mean, there's huge problems with it, and only when you sort of accept that if you adopt what Mountcastle said and say, yeah, there's got to be something similar going on here, then you can adopt both. You can say, okay, V1 is learning complete sensory motor representations, and so is some other region. And yes, but they're not exactly the same representations, but they're both doing the same basic thing. If you argue, well, that's not an efficient use of resources, that's not a good argument in my mind. We have to accept what biology presents with us. And biology is the biology is presenting with us neocortex. It looks like neocortex. It doesn't look like, visual cortex is completely different than auditory cortex and so on. So we have to accept what we're presented with, and that's how you go there. If you just say, well, I think it's more efficient to divvy up the work this way, well, fine, but it's not necessarily what the brain is doing.
I'm saying that the work needs to be divvied up somehow. So maybe I'm misunderstanding it, but well, if you have like very similar models of, let's say, a cup in ACC and in V1, what are you gaining from that?
First of all, I don't think we didn't claim that in
right. I'm just sort of trying to picture that.
Yeah. So the idea is that you're going to have many models of cups, but they're not everywhere. And honestly, we don't really understand exactly what's going on in the hierarchy yet. I think we've made that clear. Now in the book I'm writing, I've made that clear too. We just really don't understand what's going on in the hierarchy yet. So there's multiple hypotheses we could work on. One is that you could be saying, well, V1 obviously cannot represent very large, complex compositional objects, and so you could be arguing that what V1 is sending to somebody else is not a feature, but is a subcomponent, like your wheel on the Segway or something like that. So that compositional objects may be represented more complex compositional objects. That's one hypothesis. Another hypothesis is that you're changing the reference frame. Clearly, V1 is going to be working in a retinotopic reference frame, but when I get to higher levels of the cortex, I'm working in other reference frames. And so you need a hierarchy to move between these reference frames. There's lots of possibilities. I don't know yet the answer to that, but it doesn't take away from the idea that each region is doing fundamentally a sensory motor inference and modeling objects based on a reference frame. They can be just very different reference frames and representing different objects. So it's just that what's not going on is passing features between regions. You're passing already composed objects.
I agree with that absolutely.
And I just want to interject a little bit. I'm conscious of the time. It's about it's almost 11:40, so it's been about an hour and a half roughly. So we should be wrapping up in the next few minutes.
Right. So the last thing, the thing I wanted to get into was how does anyone have any alternative ideas or arguments against this idea that
I have?
Yeah,
I have a question on the first question. A question/comment. Even our biggest networks today, they're very tiny compared to the brain, right? So the capacity of our largest networks are very tiny compared to the brain capacity. So in machine learning, deep learning, the literature on continual learning focus on this segregation issue because you want to fit a lot of things into a very small network. While in the brain, we have this huge capacity, and it's my understanding, and I could be wrong, that we could learn a big chunk of the microscopic world we observe, we interact during our lifetime, and the savants are one example of that. And the question in the brain is different from how do we segregate. I think the question is how do we generalize? Because I think we have capacity to segregate everything, but then we have to generalize. While in our networks, we can generalize well, but we have this tiny capacity, and we have to find out how we can learn different things using this tiny capacity. So I think the question would be different for deep learning and the brain, but that's just my view of it.
So sorry, go on.
The way that people are doing segregation in deep learning, I think, is very different than the way segregation is occurring in the brain. I think that's a correct observation.
One other thing that we've always noted is the fact that compared to neural networks in the brain has extremely large horizontal aspect to it, and maybe not nearly as much depth. So in that respect, what we're trying to do with achievable neural networks hasn't really modeled some of the aspects of the potential aspects of what's in the human brain, other than maybe the stuff that Ray neuromorphics might be doing sometime in the future. So it's a question of whether that's how much of that is a qualitative difference, and what kind of things you achieve and what you do to your capacity when you go sparse but very, very horizontal.
Yes, that's true. I have one little thing that I can mention about the generalization issue. This is something we've discussed internally at Memento, but we haven't published. I don't think it's in any of our papers, but it's an interesting idea, and I think there's some truth to it. When you're presented with a novel object like the one-wheeled motorcycle you showed, Aris, you see something new you've never seen before. How do you know what it is? How do you make sense of it? And what I think is going on is that what happens when you see something you don't recognize, you attend to the subcomponents one at a time. So in this case, you might attend to the fact there's a wheel, and you say, oh, and there's attend to the fact there's a person, and you attend to the fact there's something that is a body that might look like a motorcycle or something like that. And the point is, you've never seen this arrangement of components before, but you keep attending to smaller and smaller pieces until you recognize what the pieces are. And now by just scanning, attending to them in order, you're building up a structure, a new object, which is composed of already familiar subcomponents, but in a new arrangement, in a framework, in a different arrangement. And what happens, I think, is going on, this is not a particularly good example, but in general, what's going on, I see something I haven't seen before. I see maybe it's got like 10 different components to it. I see 3 of those components arranged in a certain way. That's the same way it might be arranged on a motorcycle. I see another 3 components arranged a certain way. That may be the same way they're arranged in a car, and another 3 components may be the same way they're arranged in a door, or something like this. And the point is that when you see a subset of components arranged in a certain way, it invokes whatever other objects that had that also have that subset. So you can see the similarity to an object to other objects you've seen before, based by attending selectively to the individual components, in not all of them at once, but in different collections. And then what you do is you can ascribe affordances or behaviors to that object. You say, well, if I've seen 3 components that look like I've seen in another object, maybe it has the same behaviors that other object has. And if I see these components arranged in object B, it may have behaviors like that. So you, at least in terms of an inference point of view, you can look at a new thing you've never seen before and start guessing what it does. If I see something that looks like a doorknob, even if it's not on a door, I'm going to say, well, that's something I can grab and turn because I've seen that before, even though I've never seen it in this location before. We haven't developed this idea, but I think it's really what's going on as we generalize, as we try to understand novel things that we've never dealt with before. It requires attention and requires a framework in which to arrange subcomponents in a similar arrangement as you've seen in previous objects. So we've never written that up, and I don't think we've ever modeled it, but I think there's some validity to that.
What do you mean exactly by attention in this case? Like, for example, if I flashed this image before you were able to saccade.
If you flash an image like this, I wouldn't recognize it. Unless it's close enough, I might say, oh, that was a motorcycle. But if you flashed a Segway and I'd never seen a Segway before, I would have said it's a push lawnmower. So, and there's lots of things I can flash in front of you that are new, weird things you've never seen before. You have no idea what it is.
Oh yeah, but you could sort of, I think you could still, I mean, of course, there's a novelty component.
You'd only be able to, I'm a claim that you would only be able to make a guess about it if you could attend, even mentally, internally, if you could have attend to the different components, but you won't do it in a flash. You won't just sit in a flash. Take a new word you've never seen before. You've never seen a word before. Here's a long, complex word. What do? You can't recognize it in a flash. If I flash in front of you, you'll say, I don't know what that word was. Maybe it's in a foreign language. You don't know. But what you can do is then you can attend to the individual syllables, and you might be able to figure out what that word might mean based on the individual syllables. You can't do it in a flash. It's just not possible.
I'm not sure exactly what you mean with attention, because attention is kind of a vague word.
I mean, you actually narrow down the sensory input to the cortex to the one component that is now recognizable. So if I'm looking at the Segway, I can attend to the little square, the rectangle there, and I'm saying, oh, that is a wheel.
But what drives it? Is it like novelty? Is it like a prediction error? Is it?
Well, no, I said that's a wheel, and then I see, oh, there's something in the blue area that's a handle, and it looks like the kind of handle I put 2 hands on. I've seen that before. I recognize that, and I recognize wheels. I see 2 wheels, and now I see 2 wheels in a certain arrangement. I say, yeah, 2 wheels like that in that arrangement is typically on a single axle. If the other wheel was at some weird arrangement, I would say, I don't get that. I don't know what it is. So 2 wheels in a particular arrangement, as I tend to one and I tend to the other. And as I do that, I put them in a framework and say, this is their relative location to each other, that relative pose. That's what I'm talking about. It's a narrowing down of the input to the point where you can then recognize what that subcomponent is.
So you're segmenting it.
You're segmenting it into parts that you recognize. If there was nothing recognizable in that image, it would just be a blur and you couldn't do anything. You just keep getting smaller and smaller until you recognize some component. And you say, okay, that is, I got that part. That's a nut. That's a wheel. That's a whatever. Or you might say, oh yeah, that's an axle with 2 wheels. Whatever you can recognize. That's something you previously learned. I believe this is how we learn everything in the world. We attend to components, and we talked about this in the method. I'm not making this up right now. We've talked about this quite a bit. You can attend to components, and you build up an arrangement of them, and to the point where you say, yes, that arrangement is something I've seen before, even if it's not the entire thing.
Right. So okay, I thought what you meant was like there's necessarily a sequential component to this, like first wheel. But if you have like a couple things, in the same image that you recognize, this would have to happen in parallel. So like in this case, attention would be like a distributed co-occurring process.
No, I think attention is you recognize, if you don't recognize the entire thing, you literally have to start subsequently attending to some component of it. That component could be another complex thing. Take the coffee cup example, right? Maybe I haven't seen that coffee cup with the mental logo before. So I see that's a new coffee cup. Then I attend to the logo. And I say, oh, I recognize that. That's the Numenta logo. And all of a sudden, all the components of the Numenta logo come into play, and you go, I know what that thing is. It's now relative to the coffee cup. I got the new object. I don't need to look at the lines and the circles and all that other stuff. If I've never seen the Numenta logo before and I've never seen words, I would have to look at all those little components one at a time to try to build a new representation.
I think rather than the word segment, partitioning would probably be a better thing, because segmentation has connotations to it.
Right. Yeah, it's more, you're doing this in like 3 dimensions. You're attending to component at some pose. You're arranging them in a way, and you're saying, and then that would invoke the memory of other objects you previously learned that have those components in that arrangement.
It sounds like you're describing more of a recursive process than a sequential process.
What process?
Correct to infer that?
I heard the recursive.
It sounds like you're describing a recursive process as opposed to a sequential process.
Well, I don't know what it is sequential in time.
Yeah, I mean recursive as you go deeper and deeper until you find something you actually recognize, and then you go.
Yeah, you could call that recursive, but it's basically you dig deeper and deeper and deeper until you recognize something. It's not like, a recursive could be like, I recognize the mental logo, but I can still attend to the components and look at them individually. But usually I wouldn't do that. It's a good question what the right terminology is. I don't know.
Yeah, I'd be really interested in talking about this at some other point.
In maybe 10 minutes.
I need to head out
in
now.
My shower right now.
Yeah, okay. I need to head out now.
Talk about
it's about
something?
11:50.
All right. Well, why don't we? I know we've gone long here, so why don't we end it? We're supposed to have a lunch meeting here anyway.
Yeah.
We should have conversations about this and try to get everyone up to speed at least on what we
absolutely
think we've learned so far.
Cool.
Okay.
Well, thanks, guys.
Thank you.
All right. Thanks, everybody. Okay. Thanks for watching the stream. So I gotta go because I've got a meeting at noon. I'm not sure if there's going to be a Wednesday stream or not, but if there is one, I will be streaming it. So take care, everybody. Please don't forget to give the video a thumbs up. Thanks for doing that, and subscribe to our channel. Already got 16 likes. Bunch of viewers this time, so thanks everybody. If you're new for watching, we stream pretty much weekly, although I had to take a week off, but back at it for the time being. I am ending the stream, everybody. Take care out there in internet land. Stay safe.